system to produce immunity against specific pathogen. When you talked about vaccination, vaccination is an act of injecting, someone, with to be able to protect the person from, to stimulate the immune response of the body. Like we have in the north, all these polio viruses, we have a lot of, organisms, a pathogen that attacks human being. It could be virus, it could be bacteria that attack an individual. So we give vaccine to kill or to weaken those, pathogens in the body systems, or to cause the body's immune system, to stimulate the body immune system to be able to fight against those incoming organisms. To be able to explain and define what is vaccination. And then we have types of, vaccines. We have live attenuated vaccine. I may ask a question that says, define vaccination and its role in disease preventions. Define vaccination and discuss what are the role of vaccination in disease condition. So you need to define what is vaccination. You need to tell me the principle. You need to state the principle. How does it work? You can briefly mention type of vaccination, and then you talked about immune response. We have several immune response. We have natural immune response, like as an individual. There are white blood cells in our body systems. What they do is to fight against incoming organisms, and what the vaccination does also is it can help to stimulate those immune systems in the body systems. And we have what they call herd immunity. Herd immunity is a vaccination protect individual who receive the vaccine and contribute to community immunity. What it means is that, like in a class now, in this class, we have 63 students, and only 50 students are attendance now. They are listening to me. If the 50 students are being immunized, in the class, it is called herd immunity. It will not allow those, infectious disease to spread equally or evenly, among the class. That's what it means by herd immunity. When some group of people in the community are being immunized, it inhibits others from being infected from such a disease. That's what it means by herd immunity. Okay. So also we have a type of vaccine. Vaccine can be made from different types of antigens, such as live attenuated vaccine, inactivated vaccine, subunit vaccine, and messenger RNA vaccine. Each type of vaccine works by presenting the immune system with specific antigen to stimulate an immune response. Vaccine schedule, global impact, safety and efficacy. But the major thing I want us to put attention, to pay attention is preparation of vaccine. How is this vaccine being prepared? How is vaccine being prepared? We need to pay attention to the purpose. How do we need to purify vaccine? We need to pay attention to this preparation of vaccine. Number one is research and development. That means you need to identify the target pathogen. You design the vaccine. Which type of vaccine you want to produce? Scientifically design the vaccine based on the characteristics of the pathogen. It is polio. What is the characteristic of polio? You need to select the particular diseases and appropriate vaccine. You design that vaccine according to what diseases you are looking to, or infectious diseases or pathogen you are looking at. Number two, clinical testing, clinical trials. We have phase 1 trial. The vaccine is tested in a small group of 18 volunteers to evaluate safety, dosage, and immune response. Phase 2 clinical trials. The vaccine is tested in larger group, further assess safety, immunogenicity, and then optimal dosing. Phase 3, that the vaccine is tested in thousands of individuals, evaluate efficacy and safety of potential adverse effect under real-world conditions. Then regulatory approval is like just like that. They have to look at it, test it, whether it's okay, or in case of those such a vaccine produced. Then manufacturing, they will have to plan large-scale production of those vaccines. Then in the factory pharmaceutical company, they do formulation of the vaccine. The quality control test each batches, the efficacy or how to meet the regulatory standard. Also, distribution and administration, storage, distribution, administration are key thing in the distribution administration. Then monitoring or surveillance. So preparation of vaccine is very, 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 very important. All right. Then purification vaccine. Why do we need to purify vaccine? Very important. You must know why we need to purify vaccine. The purification of vaccine is a crucial step in the manufacturing process, ensuring that the final vaccine product is safe, effective, and free from contaminants. Purification methods are designed to separate and remove impurities such as proteins, nucleic acid, cellular debris, and other components. We want to look at the overview, general principle of STEM involving purification. 1, harvesting and clarifications, separation and fractionalizations, chromatography, ultrafiltration and diafiltrations, precipitations, purification and concentration, stabilizer formulation, quality control and testing, sterilization and filtration, fill and finish. So, is then also in the fasting alone, almost 3 questions going to come out from fasting. So go through the fasting and then you are good to go. Then I talked about the monoclonal antibodies. How you will remember? You still remember monoclonal antibodies, monoclonal antibodies. But pay attention to monoclonal antibodies, and then that is what that is all in 406. Okay.
Thank you, sir.
Okay. So that's what I missed out in my revision. Vaccine. Read your note on vaccine. Vaccine. So an interesting topic is, and most of us are like Dr. Kenny. If it doesn't read, we just have A, because I know that is his own area of specialty. So just pay attention to that fascination, and then there we go. Thank you. So please let me inform Mr. Fasasi. Mr. Fasasi.
Yes, sir.
So anybody that have those lecture notes can still be shared, because I saw someone that was asking about the lecture note on what I've taught. Thank you.
All right, sir. Thank you.
We appreciate you, sir.
God bless you.